Hi there. So today I wanted to talk to you about lipid nanoparticles for drug delivery. If you're wondering what that means, then here's a little picture of a liposome, and we'll get more into that in just a minute. So what are lipids? Well, lipids are naturally occurring organic compounds, and they're either hydrophilic or amphiphilic. We're going to deal only with the amphiphilic ones today. It's really a big category of compounds, and it includes fats, fatty acids, waxes, mono, di, and triglycerides, fat-soluble vitamins like A, D, E, and K. They make up the cells in your body, they're in the food you eat, and they're in the medicines that you take. They're also, when I say they're in your body, they're in your cell membranes. And of course, the cell membrane protects cells from their environment. And the membrane, as you probably remember from middle school and high school biology, is selectively permeable. So desired substances like organic molecules and ions can move in and out through the walls of the cell membrane. But it keeps the stuff that you don't want out. Cell membranes are made of phospholipid bilayers with embedded proteins. So phospholipids are pictured here in this little cartoon, and they have a hydrophilic head, usually with a phosphate group there, phospholipid phosphate, and then they have this hydrophobic tail, actually two little tails, that are made of fatty acids, so they trail off the side. Now this is an amphiphile. It has a water-loving end, and it has a water-fearing end, a hydrophilic and a hydrophobic side. And so what happens is they self-assemble in aqueous solutions, and they self-assemble into these bilayers. So if you're in an aqueous solution, the hydrophilic heads will stick out towards the water side, and then the hydrophobic tails will turn inward towards one another. So they have this bilayer kind of structure. And the bilayer structure can be in the form of a membrane, as is pictured here, or they can wrap into sort of spherical or oval type shapes. Okay? So what happens is that they end up looking like this. And when they have this bilayer sheet that wraps around in on itself into sort of a spherical shape, that's called a liposome. It's called a micelle if it's not a bilayer, and you just have the water-loving head sticking out towards the water and the tail sticking in. So some of these shapes are perfect for encapsulating drugs, right? Like vitamins or cancer-delivering drugs, and this works for nanoscale drug delivery systems. The liposomes, as you can see, have an aqueous core, so you can imagine that you could mix the drug in with the aqueous solution, and then the drug in the aqueous solution would get encapsulated in the inside of the liposome. Okay. There's also something called the niosome. They're structurally similar to liposomes in that they form a bilayer, but they're made of non-ionic surfactants and cholesterol, things like that, and they usually only have a single hydrophobic tail. Liposomes are made of phospholipids, and they may or may not contain cholesterol. Phospholipids have the two tails. The niosomes are usually more stable, as the phospholipids and the liposomes are prone to oxidative degradation, and niosomes are also inexpensive and easier to make and store. However, I'd like to point out that it's the liposomes that are most commonly used in nanoscale drug delivery systems currently. Usually what's happening with these other kinds of compounds that are coming up is they're still being tested. Liposomes have been around for a little bit longer. Okay, so here's a little schematic of everything that could be going on with these liposomes. So you put like I said earlier, you put the hydrophobic drug and they get inside the bilayer of the liposome, and then the hydrophilic drugs are encapsulated on the inside in the liquid. Okay, so you can have either hydrophobic or hydrophilic drugs, and all you do to mix the drug in is you just mix it in either the organic solvent that you put the liposomes in when you start off with. You can put hydrophobic drugs in there, and then they'll get encapsulated into the membrane. Or you can mix your hydrophilic drugs in with the aqueous solution, and then they form and they get inside. So liposomes are safe for drug delivery systems. Basically, they're made of the same kind of stuff that makes up our cells. And so you can imagine that they're biodegradable, they're biologically inert, they're non-toxic, and they're non-immunogenic. Okay? So that means that they're pretty safe for use in drug delivery systems. And they're currently, like I said, liposomes are currently the most common vehicle for targeted drug delivery that's on the market. Now, targeted drug delivery is really useful for diseases like cancer, okay? If you remember, until recently, cancer drugs would just go throughout the system when you took chemotherapy, for example, and they would kill both healthy and cancerous cells and cause serious side effects. Targeted drug delivery uses molecules known as ligands, for example, which attach to the outside of the nanoparticle, and those ligands will bind preferentially to molecules or receptors on the tumor cell. And this enables the delivery of the drug to the cancer cell, and it's not then delivered to healthy cells as often. So it cuts down on the side effects. For example of targeting, it's known that folic acid receptors are overexpressed in many types of cancer cells. And so, for example, you could attach a ligand which wants to bind to these folate receptors in cancer cells and target the cancer cell in that way. Because it's known that the folate receptor density increases as the grade of the cancer worsens. So you get your cancer cells and they have more and more of these folate receptors than does a healthy cell. Okay, so the drug with the receptor, the ligand that targets the folate receptor, would be taken up by cancer cells more than it would be taken up by regular cells, and that would help reduce the side effects of delivering a cancer drug to a tumor cell. There's other things that you can do. You can see here in the little cartoon there's these sort of brushes attached to the outside of the liposome. A lot of the liposomes that are on the market currently have polyethylene glycol attached to the outside of the liposome. And that's added in a process that's known as pegylation, because they abbreviate polyethylene glycol as PEG. So PEG prevents the particles from binding to proteins in the blood and attracting the attention of the immune system, and it can aid in allowing the drug to stay in the system longer without being broken down because it's not being attacked by your own body. Now, liposomes are already on the market, and then there's a lot more that are in clinical trials. For example, there's a drug named Doxil, which is pegylated liposomal doxorubicin. And it was FDA approved in 1995, so it's been on the market a good long time now. And it's being used to treat ovarian and breast cancer. So doxorubicin was already approved to treat cancer. It was a drug that was already known and used to treat cancer. However, there were serious side effects to doxorubicin. It could cause severe and life-threatening heart problems, sometimes even years after you took the drug. So instead, if you give it in a liposome, it has fewer side effects because the dosage of the drug can be lower and you can target it. There's also a drug on the market called Myoset, non-pegylated liposomal doxorubicin citrate. It's been approved in Europe, but not the United States. There's another cool example that's currently in clinical trials, and that's a temperature-sensitive liposomal doxorubicin. And what happens there is that the liposome has been engineered to break down at temperatures between 40 and 45 degrees Celsius. So at those temperatures, the liposome wall breaks apart, and then it releases the drug that's inside. Now, that's at a temperature kind of like running a high fever. But what you can do is you can use radiofrequency ablation and target the heating only to the cancer cell. What they do in radiofrequency ablation is they insert a little needle into the tumor, and then they pass an AC current through it and makes it emit radio waves. And those radio waves heat up the tumor to sort of that fever temperature, okay? And then that causes that liposome to break apart and release the drug only in the spot where you need it, only in the spot at the tumor. This is a possible treatment for liver and breast cancer, and it's in clinical trials now, like I said. Perhaps in the future, it would be nice too, instead of radiofrequency ablation where you insert the needle, to use something like focused ultrasound to heat up that tissue. And then you could heat the liposomes non-invasively, so maybe that's a possibility too. There's other delivery systems that are currently on the market, or currently being tested. Newer structures such as solid lipid nanoparticles and nanostructured lipid carriers and nanoemulsions have also found to be better performers than liposomes. In particular, the nanostructured lipid carriers have been identified as a potential next-generation cosmetic delivery agent, and that can provide enhanced skin hydration, bioavailability, and stability of the agent, and controlled occlusion. So the encapsulation techniques have been proposed for carrying cosmetic actives, and nanocrystals and nanoemulsions are also being investigated for cosmetic applications, and there's some that are already currently on the market. When I say cosmetic applications, you may be wondering what the heck I'm talking about. Well, it turns out it's for face creams, for wrinkle creams. Maybe I should buy something. What do you think? There's some on the market right now that are using these kinds of nanoparticles to encapsulate vitamins like retinol, vitamin A, vitamin E, and C, and then you rub it in the skin cream. It gets taken up and released and has shown to be good. Particular retinol, for example, has a tendency to break down or degrade. It has a short shelf life because of if you expose it to ultraviolet light from sunlight or whatever, it'll break down. And so if you encapsulate it in the liposome or some sort of nanoparticle, then it's protected and it doesn't break down as quickly, which increases the shelf life of the retinol. And of course, retinol has been shown to fight wrinkles and aging. Okay. So there's lots of particularly vein and shallow uses for nanoparticles, which will probably make a lot of money, in addition to sort of the altruistic applications of cancer fighting. Now, if you want to make a liposome, it's actually not too bad to do so. The commercial fabrication of the liposomes is done to specifically select certain sizes, but just making liposomes in general isn't that difficult to do in the lab. So one way, for example, to make a liposome is to dissolve your lipid in an organic solvent. Remember, these things lots of times have the hydrophobic end. So if you want to dissolve it nicely, you usually dissolve it in some kind of organic solvent. And then what you do is you pump the organic solvent off or dry it out in some way so that a film of the lipid is going to line your container walls. So you'll have a sort of a solid biofilm on the walls of your container. And then you pour in your aqueous solution, including your drug, for example, and you swirl it around or agitate it in some way. And then what happens is that the lipids will naturally just kind of come off the walls, and then in the presence of the aqueous solution, they'll self-assemble into the nanoparticles. So that's a really cool thing. The liposomes will self-assemble. Now, if you want to control their size after you initially form them, then you can sonicate or emulsify or extrude them in some way, and then make the ones that you've already made break apart and reform again, and break apart and reform again. And when you do that, you can successively decrease the size of the nanoparticles that you've made. There's another easier way. It's just that it doesn't give a lot of high quality liposomes. You can just mix it, right? You can just mix your liquid with your aqueous solution straight off, okay? And then they will self-assemble into liposomes. However, the quality isn't as good. There'll be more defects and they'll be bigger. Okay, so if you want to, for example, I've seen YouTube videos of people making liposomes in their blenders at home or with ultrasonic jewelry cleaners, where they just mix their lipid with whatever they want to do. Lots of times it's vitamin C. If you Google how to make liposomes on YouTube, you'll see a lot of videos about that. Mix it with the vitamin C in an aqueous solution, liposomes form. There you go, Bob's your uncle. You got yourself a liposome. All right, well, I hope you enjoyed that discussion of self-assembly of liposomes for targeted drug delivery. And as always, if you have any questions, let me know, and I'll see you next time.